Hello guys, let's talk about a new topic today, that is a stem cell. Stem cell is nowadays a very common word used both for the plant and animal. But today we'll be focusing more on the animal because the plant cells has a lot of totipotency and the plasticity. The cells can be molded in any way. But today's topic is mainly concerned with the animal part. Let's talk about it. So those cells we call a stem cell, which is unplanned or unprogrammed, or maybe with the time they get little planned. So we can start with a range of those cells, right from the totipotency to pluripotency to multipotency, and finally we jot down to the unipotency. So these stem cells are the one which are unspecialized. They do not know exactly which way they have to develop. They have only ability to divide on and on and on with a lot of regulations, and later on they get differentiated into a particular type of cell, or maybe several type of the cell. If it is a pluripotent, nearly 200 types of the cells it is going to make. Now what is going to be the fate of a stem cell? We'll talk about this in little more detail in a while. Stem cell, either they fail to differentiate or undergo the apoptosis or the neoplasia. Another way it can differentiate, then the senescence comes and it will undergo the apoptosis. And the third thing, it has the ability to make its own number, that is called as renewable capacity. This replenish the old and damaged tissue by proliferation and maturation as well. And keep in the mind, they are going to maintain their number as well. Now, the places where we have the stem cell, those are called as stem cell niches. And these niches has a signal which will help these cells to do the three things. One, they maintain their own number, that is called as self-renewable capacity. They get a signal to survive. Third thing, they have a maintenance role to play as well. For example, if I tell you the one simple example, you see in the case of intestine, where we have the crypts of liver and all, there we have a cell we call a Paneth cell. Those Paneth cell, they release the WNT, or we call as a vent, to increase their number. And as the cells cutting off and going away from this WNT signal, they are going to get differentiated. So here the niche cell, basically they are their stem cell by cell to cell addition having the specific protein, and where they get all the signals to maintain their number, to differentiate as it goes away from this. Let's see a very simple example in order to understand this. If I have the fertilized egg, and this fertilized egg is called zygote, and we know all the cells, all the organs come from these cells only. So they are having totipotency and a level because of blastocyst will form. You can see inner mass of cells are shown over here, and the trophoblast are on the surrounding. This is the time when this is going to be implanted. We can take out the cells from there, from the inner mass of the cells, and we can culture them and we can use them as a stem cell. Otherwise, those are these cells are going to differentiate in the different kind of cell. For example, they can make with the hematopoietic stem cell, they can make with the neural stem cell, we can which can make with the mesenchymal cell. Blood cells is going to be formed from the hematopoietic stem cell. Neural stem cell give rise to glial cell as well as the neurons, whereas the mesenchymal cells will give the connective tissue, bones, and etc. Then we have the pluripotent stem cell, which has huge ability. Just now I shown you, and all the different kind of cell lineages, roughly around 200 plus kind of cells can be developed. Now there are the few basic properties of that. The stem cell may undergo the symmetric cell division. That means the cytoplasmic content is going to be same in the daughter cell as well. Whereas it could also undergo the asymmetric stem cell, which you can see in the case of the second. And third will give rise to progenitor. As it goes further, the totipotency level decreases. And finally, you can see the fourth one, which is terminally differentiated. You can see the A, B, C, all the scenes. A is going to be stem cell, B is a progenitor cell, and C is going to be differentiated cell. Now, the main point which scientists look at, and you can see the name of the scientists, Takahashi and Yamanaka, Sanya Yamanaka, who got Nobel Prize as well for creating the induced pluripotent stem cell. So once they studied these stem cell, what they could understand is that there's a certain group of genes which are playing important role. They initiated with around 24 different proteins, and they have categorized them into the three categories: group 1, group 2, and group 3. Group 1, which is the specific transcription factors, they're very, very important in the early embryonic stem cell to maintain the pluripotency. Then the proto-oncogene cells, which are very, very important for the cell proliferation. You can see the few example over here: TCL1, STAT3C, make ERAS, KLF4. And the group 3, which is not known too much, but yes, they do play important role in the certain processes. Now let's talk about the few important proteins. For example, the OCT3/4, which is involved in the maintenance and the self-renewal of the pluripotent stem cell, and which is very, very important. Like, for example, if you see the trophectoderm, where this is going to be repressed, and overexpression will always lead to the various lineages of the cells. SOX2, which is essential for the embryonic development, and down regulation in this case is going to be done by the siRNA silencing. NANOG, it is another embryonic stem cell along with the the protein NANOG along with the OCT3 and 4, and SOX2 is very necessary to maintain the pluripotency. One more protein, that is a messenger RNA binding protein, we call the Lin28, which express in the embryonic stem cell and the embryonic carcinoma cell associated with the differentiated and the proliferation. Now, if you see this, what you come to know is that the pluripotent stem cell, they differentiate to make the primitive endoderm and the mesoderm. They can maintain the self-renewal number, because we know today every organ has its own stem cell, because other cell, they have a limitation, they cannot divide for a long due to the Hayflick limit, the sorting of the telomere. That is one of the reasons. So stem cells are going to be very important, and usually they have the activated telomerase, so their sorting of the telomere will not occur in this case. Hence, the stem cell can be put to the many uses. Now, the science has gone to the level where we can convert the differentiated cell into the stem cell. Those are called as iPSC, induced pluripotent stem cell. This stem cell can be used for treating the several diseases, right from the baldness, blindness, deafness, on the other end, the brain stroke, traumatic brain injuries, Alzheimer, Parkinson, missing teeth, then amyotrophic lateral sclerosis, myocardial infarctions, muscular dystrophy, diabetes, multiple site cancers, bone marrow transplantations, spinal cord injuries, rheumatoid arthritis, so on and so forth. So they're quite useful. The reason is that certain region which is, if it is, damaged, so we can use a stem cell to control them, and we can, regenerate the new organ for that. Hopefully you enjoyed it. Thanks a lot for listening patiently. Kindly subscribe the channel for more new important topics which could help you in the future. Thank you very much.